Today I'm presenting my presentation entitled Considering the Interaction Between Crop Residues, Bioeconomic Conversion Pathways, and the Return to Carbon to Soils. This is part of my PhD project, The Sustainable Role of Agricultural Residues in Future Bioeconomy Strategies and its Dependency upon Carbon Returns to Arable Soils. I'm doing my PhD in the National Institute of Applied Sciences, INSA, in Toulouse, France, with cooperation of the Technical University of Manabí in Ecuador, under the supervision of Dr. Laurie Hamelin and co-direction of Dr. Ezequiel Zamora and Dr. Ariane Albers. This project has been funded by the French Embassy in Ecuador under the FSPI project and by the French National Research Agency under the Make Our Planet Great Again project. I started my PhD on late March, beginning of April this year, 2020. So why this study? Due to its composition and wide availability, agricultural residues are fixed for a variety of bioeconomic conversion pathways. However, these residues have normally been left on soils as a way to maintain the soil organic matter. Therefore, the removal may reduce the soil organic carbon content, decreasing the soil quality and causing other environmental issues. In fact, the Intergovernmental Panel on Climate Change has recognized the importance of considering the impact of using these biomass resources for bioenergy. Some authors have tried to determine which is the amount of residues that could be harvested without affecting the soil quality. Yet, the availability of the agricultural residues depends on specific local conditions as soil quality, climate, farming practices, and the sustainable amount that can be removed is especially explicit. If we adopt a universal or too conservative threshold, we may deprive bioeconomy and bioenergy from an important feedstock. Bioeconomy involves conversion pathways where we obtain co-products that are rich in carbon that is more resistant to degradation. This carbon could be returned to the soils as a way to maintain or enhance the quality. So optimizing the returns to the most carbon vulnerable soils can play a role in enhancing the sustainability of using these crop residues. The aim of this work is to develop a spatially explicit understanding of the amount of straw that can be harvested from agricultural soils for a variety of bioeconomic conversion pathways, while we maintain or enhance the long-term stocks compared to a reference situation without bioeconomy for the whole territory of France. We consider the long term as a period of 100 years, and we are calling the crop residues as straw because these are the main residues we are considering in the study. So what's important to understand in our work is that we are going to determine the amount of residues that we could harvest in each bioeconomy scenario we are considering and compare it to a reference situation where there is no harvesting for bioeconomy. So we could have a relative vision. We have two main research questions. First, how much straw can be harvested to be used in each bioeconomy scenario while we maintain or enhance the current soil organic carbon levels? And to which extent the redistribution of the carbon to the most vulnerable lands will affect the sustainable level that we are determining with the first question. So our hypothesis is that the conversion of agricultural residues and bioeconomy processes, followed by the return of the bioeconomic co-products to the soils, will allow to harvest a higher amount of residues, then allowing a higher production of energy without affecting the soil organic carbon levels in the arable lands. This graph explains a bit our vision. So the residues are taken to the bioeconomy conversion facility, and after the process, the co-products, which are rich in carbon, are returned to the soils as a way to maintain or enhance the soil levels. The bioeconomy conversion pathways we are studying are 6: anaerobic digestion, pyrolysis, hydrothermal liquefaction, gasification, bioethanol production, and the production of bio-based products. We are still considering and selecting which products we are including in the bio-based products pathway. So as I said, we are focused on the co-products. So for the anaerobic digestion, we consider the return of the digestate. For the pyrolysis, the biochar. For hydrothermal liquefaction, the hydrochar. For gasification, both tar and biochar, depending on their composition. And in the bioethanol fermentation, both the vinasse and the lignocellulosic fibrous residue. So we have to determine through mass balances the amount of carbon that we could return to the soils, and also we need to determine the degradability of this carbon. Our project is divided in 4 different stages. So the first stage is to determine the amount of straw that we could harvest when we consider the return of the co-products to maintain the soil levels or enhance them. Then we are going to optimize this through the redistribution of the carbon to the most vulnerable lands first. We are going to perform sensitivity analysis on the stability of the co-products, the climate change, and other factors that could affect us. And at the end, we are going to extrapolate this methodology to a static case in a province in Ecuador, the province of Manabí. Since I'm in an early stage of my project, today I'm going to present the methodological approach that we are following to complete stage 1, which I'm working with now. So to complete this first stage, we are following 3 steps. First, we cluster France, following the type of climate, soil, and farming. Using geographical information systems, we have gotten climate, soil texture, and the initial soil stocks data. This information, we cross it with the crop rotation and farming practices in the whole territory of France to create what we call our simulation units. Now we are performing mass balances for each bioeconomy pathway that we are studying to determine the amount of carbon that we will be returning. And then we are going to use a SOC modeling software. So considering the simulation units created and the carbon that will be returning from the co-products, we are going to determine the long-term SOC levels. But what we actually have to do is to model until we obtain long-term SOC levels that are equal or higher than the initial SOC stocks. And then we will obtain the amount of residues that we could use for each bioeconomy pathway. So for the first part, we are working with collaborators from INRA France, and we are basing our project on the data from the 4,000 report. So I'm presenting this methodology from Polari et al. 2019. What we have done to create our simulation units is to cross the climatic information in the saffron grid with the soil information in the cartographic units to create the pedoclimatic units. Then these pedoclimatic units has been selected using the agricole parcel register, where each farmer uploads information about their crops to select only the UPCs that are agricultural. Then we have selected only those UPCs that contain the main cultures in France. So for example, we are removing those grasslands that are not agricultural. In each UPC, we have many different crop sequences, but only the main 3 sequences have been selected. And also for the type of soils, we have selected only those that cover 70% of the UCS. Mixing all this information, we have 62,557 simulation units. So this map presents the units that we are using. The red ones are the UPCs that contain the main agricultural crops in France. So with this methodology, we have 1,591 crop sequences. When we consider the land management, we are considering 11 different crops, 3 types of land management, organic and mineral fertilization. We are modeling based on a future climate scenario, whether the RCP 4.5 or the RCP 6. And our initial sock stocks come from the national map on the 0-30 centimeters. Now, regarding the bioeconomy conversion pathways, we have to determine the mass balance. So as an example, I present in the biochar case. So based in the data from Razar et al. 2020, for a pyrolysis of 3 minutes at 400 Celsius, we consider a 45% content of carbon in the feedstock, and they determine a conversion rate of 21% into biochar, which contains 55% carbon. So it means that 26% of the carbon in the feedstock is present in the biochar. Then we need to determine or understand the degradability of this carbon. So we are doing a literature review, but this is one of the main limitations that we have because there is little information and there are not many long-term field experiments to get this information. As an example, we have here the IPCC guidelines that based in this formula, this factor is the fraction of carbon in the biochar that will remain stable for 100 years. They have defined it as 80%. So as I said, we are doing a literature review to consider different feedstocks because we are going to use this for the sensitivity analysis. Also, we are doing it for all the bioeconomy conversion pathways considered. Then we are going to model using the SOC models. So based in a study reported here in France, AMG had the best performance for the French agriculture. So we are using AMG, and we are using a second model, the ROTZ, to validate our results. Both models are very similar. So the fresh matter is split into different pools. The fresh matter is split into an active compound based on the humidification coefficient, and then a part of this is mineralized over the time. And the part that does not go to the active compound is directly mineralized as CO2. ROTC is very similar. What happens with these models is that they have been created for agricultural residues, and they do not contain factors to consider these other external organic matters. So we are modifying the models to consider the recalcitrants and the different humidification coefficients of the co-products we are using. We are doing our work using R because it's a heavy amount of data. We have more than 62,000 units in 100-year periods. It's more than 7 million rows, and AMG works in a line per line crop format. So we have transferred the information from the 4,000 project in INRA, which is based on sticks, to these models using R. Our scenarios, as I said, the first scenario is the reference that contains the current practices under a future climate scenario, which will give us the long-term stock levels when we don't have bioeconomy. And we will compare this with the bioeconomy scenarios when we return the digestate, molasses, biochar, agrachar for this future climate. Our expected results are first to create this literature review where we include the overview on the co-product recalcitrants, then to have the amount of residues that could be harvested when we consider the return of the conversion pathways to maintain the stock levels on the arable lands in France. We will also have the long-term sock stocks for the whole territory of France under each scenario. And we will have the sock variation in 100 years when we consider the bioeconomy scenario and the reference scenario. This is a reference and my information. Thank you very much for your attention.
Muchas gracias, señora Andrade. Thank you very much. Other questions?
Okay. Tell me.
Yes, we have a question.
Okay.
How far do you think your results can be generalized, transferred to other countries?
Yeah, mostly the methodological approach will be transferred because we are going to use the same methodological approach, but in a context for the Ecuadorian agricultural systems. It is difficult because each country is different. That's the highlight of our work, because some works have tried to obtain a value that's universal, but it's not very good because each country has different climates, soils, and farming practices, so the results will be very specific for each one. However, countries that are very similar to France could have similar results, but what we are going to transfer to other countries is mainly the methodological approach, and considering that when we return these co-products, we could have a higher harvesting of the residues and promote more bioeconomy processes.
You stated that you will reallocate the residues of the bioeconomy approach to vulnerable land. What do you mean with vulnerable land?
Yeah, we have under our research group, we have determined what are the marginal carbon lands in France. Those are the lands that contain less than 50 tons of carbon per hectare. So we are going to relocate the carbon to those places first as a way to enhance or maintain the sock levels.
But maybe you can find a lot of land with sand, based on sand, and heavy, and we will expect much higher carbon content on a heavy soil and with a lot of clay, and therefore maybe a clay containing much carbon is more vulnerable because you will expect there more, or this clay needs more carbon than sand soil, where it's for nature it's low in carbon.
I don't understand the question, but yes, this could be an approach considering the type of soil. But we have already selected those vulnerable lands based on the SOC map.
Yeah, it's a heavy soil type. Yeah, needs more carbon, or you will expect there more carbon. Therefore, maybe it's an approach to say or to determine that for a heavier soil you will set a higher level needed as a baseline for carbon than on a soil, light soil with a lot of sand.
Yes, that is a possible approach.
There is another question: how you did consider the use of straw for animal bedding?
Yeah, and the UPCs that we removed from the map are those that are considered for animal. We compared our UPCs map with the OTEX map, so we only left the UPCs that are not dedicated for animal growing, because if we will consider them, we won't have residues for our bioeconomy, because in the animal farms, most of the straw is used for the animals. That's why we have taken those farms from our study. We're only considering those that are agricultural.
There's a question stating that you have massive export of straw between French regions.
Yeah, that is a solid point that we are considering. There is a part of straw from some agricultural farms that is exported to other animal farms. That's true. And we have to consider that part in our study. So we are doing the literature review on data of that.
Anaerobic digestion in a feasible approach for treating the straw?
What? Sorry?
Is anaerobic digestion a feasible approach to use the straw with a lot of lignin, which is not suited at all for anaerobic digestion?
Yes, normally the straw is mixed with the manure, because it's true that the straw is very high in lignin and won't produce much biogas. So in our mass balances, we are considering the mixture with manure as it is normally produced.
Okay. Are there further questions? It seems that no. Then thank you very much for your presentation and for the discussion.